CO2 ones. Okay, dissolve oxygen, dissolve CO2. Dissolve oxygen is important because of the increasing frequency of hypoxia and anoxia. Okay, so these reduced levels of oxygen brings stress to the environment, to the marine organisms. Okay, so if you've looked at the newspapers, I mean, if you have been reading the news regularly, you will notice that there on some occasions there are reports of mass fish kills. Okay, mass fish kills where uh, almost all the fish in the cages, in the nets, died because usually because of a low concentration of dissolved oxygen. Okay, so essentially the fishes suffocate and die. Okay, so it happens uh, on occasions, and it's detrimental to the aquaculture industry. So now you know um, the main causes of hypoxia. So there can be ways to control or reduce or prevent the formation of hypoxia. So since a lot of it is because of increased respiration of organic matter that's being formed in the water body, you have to find a way to reduce the production of organic matter. So the organic matter can be from human sources, or could be in the form of nutrients from humans that enter the sea and increase the amount of primary production that inadvertently increase the amount of organic matter in the water body. So this can bring about hypoxia. Next, we covered dissolved CO2. So this is important also because of the increasing concentration of CO2 in the uh, atmosphere that is linked to climate change. So the increasing CO2 has also dissolved in the seas that has led to souring or ocean acidification that is detrimental to uh, carbonate shell organisms and also coral reef systems. So it's important to know the the factors that drive this ocean acidification. But when we learned about climate change earlier in this course, this is not one problem that this is not a problem that one nation can solve. It is a global problem. So every country in the world has to play its role in order to overcome this climate change problem. So when we talk about these, the processes of dissolved oxygen, uh, the processes related to dissolved oxygen and dissolved CO2, we always linked it to biological processes. So biological processes are also dependent upon nutrients. Okay, so nutrients drive biological processes also. So when we talk about nutrients, we have macronutrients, we have micronutrients. So micronutrients are usually trace elements. So trace elements, the one that we will cover in this course will be iron. Okay, so micronutrients or trace elements. For example, iron. But the nutrients that are taken up in a larger quantity by the marine organisms are the macronutrients. So we'll focus on the N, nitrogen, phosphorus, and silicate. Okay, so NPSI. So we now know that the uptake of these macronutrients, on average, it follows the rate fill ratio. Okay, so these are for primary producers. So the CNP ratio is 106, 16 to 1. So the N to P ratio of 16 to 1 is important here, and you should remember it, okay? Because when we are looking at the balance between nitrogen and phosphorus, we are always looking at this ratio. So this Redfield is an ocean chemist. So early on, when they had an opportunity to take a cruise around the world, covering many oceans, so he proposed a very simple experiment, just uh, collecting water from the surface and then filtering them and then measuring their uh, elements, okay, the C, N, and P, okay, so just by measuring their elements. So he was very surprised, well, after this work, that the CNP ratio is actually very, very constant across the globe, okay, with minor variations, but on average, it is always 106, 16 to 1. That means uh, for every 106 carbon that's being fixed by the primary producers, the primary producers will require about 16 uh, moles of nitrogen and 1 mole of phosphorus. Okay, so this is what they surmised from uh, his experiment. Okay, and because later on other researchers also found that the ratio holds true, they have given the ratio the name Redfield, in honor of the scientist, Redfield ratio. So why is this important? Because that means in water bodies where the concentration of N, for example, let's say in one water body, you have the constant 15 micromolar of N and 1 micromolar of phosphorus, P. So in that water body, the phytoplankton there will experience nitrogen limitation. Okay, because the N is 15, P is 1. So there is a N becomes limiting. Okay, so that is how you can use the refill ratio to understand better the environment that you are studying. In another example, if you have, let's say, 16 micromolar of N, but you have 3 micromolar of P, you will still experience N limitation, because although N is 16, the P is 3, so N will be limiting. If you have, let's say, a water body where the N is 32 micromolar, but the P is only 1 micromolar, then definitely phosphorus now becomes limiting. Okay, so that's how you can use the refill ratio to understand the the conditions of your primary producers. So when we look at nutrients, we begin to see that nutrient profiles over the water column exhibit a surface water depletion and deep water enrichment profile. Okay, so this is what it means by a surface water depletion and a deep water enrichment profile. This happens because if the element that you are studying is a nutrient, the primary producers in the upper waters will utilize it. And when it is utilized, it is reduced in concentration. As the phytoplankton dies out and precipitates, it will be degraded by bacteria. Okay. During its degradation, during its decomposition process, the element is released back into the waters. That's why you have a deep water enrichment profile. Okay, so this profile is important because let's say if you are studying an element that very few people study, we are going to the ocean, usually trace metal. For example, I know a researcher who goes, a marine chemist who goes to the oceans, Pacific Ocean, and collect clean water samples to measure cobalt, vanadium, all these trace elements. He measured them through the water profile, and then he measured them through the water column and look at the profile. So if it exhibits a profile like this, then he can safely uh, speculate that this element is an essential element, is a nutrient for the primary producers because it exhibits this kind of profile. Okay, of course, later they will carry out experiments, laboratory experiments, where they test it out to see whether it is true, whether the element is a nutrient for the primary producers. Okay, so we are doing this because primary producers fix CO2, and a lot of scientists believe that if we can understand better uh, the factors that control uh, the CO2 fixation by the CO2 fixation process in a phytoplankton, will be able to come up with solutions towards the increasing CO2 in the atmosphere. Okay, so if the nutrients exhibit this surface water depletion and deep water enrichment profile, there need to be an event that will also replenish the euphotic zone. So usually this happens when there is an upwelling or seasonal mixing. So seasonal mixing maybe during the winter time when the winds are rough, or during monsoon, will essentially bring up all the nutrients. Okay, and you will have a homogeneous concentration of the nutrient throughout the water column. Okay, so this is actually the condition before the occurrence of a spring bloom. So spring bloom, so bloom refers to the explosion of phytoplankton concentration. The dramatic increase in phytoplankton usually happens in spring season. So winter will bring up the nutrients to the upper waters. Then when spring comes, when the temperature increases, the amount of light increases over time, over day, you have a significant bloom, phytoplankton bloom. Once you have that, of course, it will slowly with the utilization of the new nutrients, the concentration of new nutrients in the euphoric zone in the surface layer will decrease. Okay, so you'll have this kind of pattern again. The macronutrients that I've stated earlier, one by one. Okay, we'll start off with nitrogen. So let's understand first the different kind of forms of nitrogen that we can find in the oceans. Okay, we have inorganic forms and organic forms. Okay, so inorganic forms, for example, your ammonium, nitrate, nitrite. Then organic forms, amino acids, urea, PON, DON. So PON stands for particulate organic nitrogen. DON stands for dissolved organic nitrogen. Okay, so we also need to understand first when we talk about uh, an element or nutrient in the water body, we need to understand the processes that contribute to its concentration and the processes that remove it. Okay, which means input and output. Okay, so the input to nitrogen are your N fixation. So nitrogen fixation, uh, it's biological. It's can be symbiotic or free living N fixes. Okay, so if you want to give examples uh, for symbiotic N fixes, you probably have your yeah, but for ocean waters, most of them are free living. Symbiotic N fixes are usually in the form of leguminous plants. So in the oceans, a lot of non-symbiotic, free-living nitrogen fixes, for example, um, Trichodesmium and Shinnecococcus. So Shinnecococcus and Trichodesmium are all under the phylum Cyanobacteria. So you have lightning. Lightning is also a natural source of N, utilizable nitrogen, ammonium. So you have anthropogenic. Anthropogenic sources comes from human activities, fertilizer, and wastewater. Okay. So in the past, anthropogenic activity uh, contributes little to the nitrogen cycle. However, with population growth, then commercial farming. So with commercial farming, in order to increase output, the utilization of man-made fertilizers increased. So this, uh, as you've learned earlier, fertilizers that uh, overuse of fertilizers will bring about runoff, surface runoff. When the rain falls, the additional fertilizers, the fertilizers that are not being used, will be dissolved into the rivers, will be brought into the sea. So this has contributed a lot of additional N to the N, N cycle. Meta-analysis of waters around the globe show that the waters in our world is slowly going towards uh, P limitation because we are producing more and more nitrogen. The concentration of phosphorus is no longer adequate to support the amount of nitrogen available. So the system is being driven towards phosphorus limitation. Okay, so other than what goes into the uh, nitrogen pool, okay, POOL, okay, nitrogen pool. We also look at what is being taken out from the pool. So assimilation, okay, assimilation by organisms. So assimilation, as I mentioned, NO3 preferable, no, ammonium preferable to NO3, but both ammonium and NO3 nitrate are favored. Okay. Then other than assimilation into marine organisms, you also have export. So exported out of the system, either by sinking, precipitation, or horizontal transfer. Okay. So once the organisms, for example, phytoplankton, is sinking, precipitating, it is being degraded, decomposed through the bacterial process of ammonification. Ammonia will be released back from the proteins and amino acids. So ammonia will be released back to the nitrogen pool. Okay, so this is essentially the input and the output affecting the nitrogen pool. Okay, so so as I mentioned, anthropogenic activities have doubled or increased the riverine supply of N. So most of the waters are driven towards P limitation. So other than N fixation, you also know that there are other processes in the nitrogen cycle: ammonification, nitrification, denitrification, and NMOX. Okay? So these are the main processes that affect the N cycle. So as I mentioned, in the microbial ecology lecture, NMOX is of course the anaerobic ammonium oxidation, right? So it occurs when the conditions when the dissolved oxygen levels are low. So, but in most oceans, you can clearly see that it's oxygenic. It's oxic. There's enough oxygen. But you must remember, microbial processes can occur at a micron level. Okay, so although ocean conditions might look aerobic, you can have hotspots, which means hotspots where there are organic matter concentration, where maybe one particle or a few clumps of organic matter. And these kind of conditions, hotspots, can create uh, anoxic or anaerobic environment that's suitable for NMOX to occur. Okay, so it's still possible for NMOX to occur. It's not it's mostly concentrated towards the hotspots. Okay, it's not distributed throughout the water column. Next, phosphorus. So we'll do the same thing. First, let's look at the the forms of phosphorus that is available in the oceans, in the waters. You have your inorganic forms, your phosphorus, no, your and also your phosphate, and also your orthophosphate. You have your organic forms, particulate organic phosphorus, dissolved organic phosphorus. So you have different forms, both inorganic and organic. So what are the main processes that will contribute the input to phosphorus pool and the output from the phosphorus pool? Okay, so you have biological decomposition and Okay. So we have both biological and chemical processes that affect the P cycle. So the main ones are decomposition. So decomposition essentially means an organism that has died is being decomposed mainly by bacteria, and as it decomposes, P is released. So assimilation is when uh, P is taken up either by phytoplankton, by bacteria, or by uh, yeah, or by other organisms. Other than biological processes, you also have chemical processes. Precipitation as calcium and iron phosphate. Okay, precipitation. So this is the basis of the estuary or the mangrove forest as the function of estuary and the mangrove forest as filters. Okay, so the general consensus is estuary works as a filter that cleans up the water from the river before it is released into the sea. Same goes for mangroves. Mangroves clean up the waters before it is released to the sea. One way it occurs is because of chemical precipitation. Phosphorus that is in the water is precipitated onto the surface of the clay. Okay, it is attached to the surface of the clay, and it can be uh, and it can sink out of the system. Okay, the same function, the same process occurs in mangrove soils. So precipitation will remove phosphorus from the waters before it is released into the sea. Okay. However, these processes is also affected by uh, the oxygen level. When it's aerobic, when it's healthy, a lot of precipitation occurs. However, when it's anaerobic, the precipitation is the reaction is reversed and it becomes soluble, and dissolved inorganic phosphorus is released back into the waters. So that's why in healthy waters, whereas aerobic, things will get cleaner with time because of this uh, processes of removal and decomposition. Okay, so decomposition and removal will reduce the amount of pollutants in the sea. However, when waters are anaerobic or hypoxic, the reverse occurs. You'll have more of this previously dissolved elements being released back into the waters. So inputs to NP pool, phosphorus pool, anthropogenic by human activities, or degradation of organic matter that will release phosphorus. The output from this phosphorus pool includes assimilation by organisms, again, lateral, horizontal export, or sinking and precipitation. Again, as organisms sink, decomposition and degradation processes occur, and this will again release inorganic phosphorus back into the phosphorus pool. So the soluble form of silicate is orthosilicic acid, or written as this. Okay. So silicate is different from N and P, okay? The main source of silicate is through the weathering process. When the when the rainwater washes over the soil or over rocks, over pebbles, over stones. There is weathering, physical and chemical weathering of soil, stones and sand that will contribute to autosilicic acid in the rivers, and this will be brought into the sea, uh, into the river mouth, and then into the sea. So this is the main source of silicate, okay, as opposed to NMP, where anthropogenic activities are also important. Okay, but for silicate is different. For silicate, it's mainly natural weathering process. Okay, silicate is also not generally utilized by most of the marine organisms, but by specific classes, especially by diatoms and uh, radiolarians, by organisms that require to form silica shell. Okay, so these are not uh, so it's not the general population, but very focused towards diatoms and radiolarians. But we learn this because diatoms are one of the main primary producers in the sea. Okay, so we need to know what controls diatoms, what affects diatoms. Okay, and diatoms require silicate to form the shells. So another characteristic that is very different from N and P is this, where the silica shell in the diatoms is only recycled back by dissolution at about 1000 meter depth. So even when a diatom dies, even when bacteria degrades it, they only degrade the organic uh, material in the diatoms. But the shell, the silica shell sinks, continues sinking at only when it has reached about 1000 meter depth through chemical, physical, and pressure processes. Dissolution starts. Okay, so it's very different from NNP. NNP, you can have in situ recycling through the degradation process of tissue material, which means in situ means when CO2 is fixed by the phytoplankton in the euphoric zone, if the phytoplankton dies, the phytoplankton can actually be recycled, can actually be degraded within the euphoric zone itself, even before it sinks, and then it can be released back into the euphoric zone. However, for silicate, it's different. It has to sink outside of the euphoric zone, far away, 1000 meter depth, before there is any chance of recycling. Okay, so that means you the only possible source of silicate in for most waters are through weathering processes and from the rivers. Okay, so these are the main sources of silicate, not not through the uh, recycling process in the euphoric zone. Okay, so this is very an important difference in relation to nitrogen and phosphorus. Okay, comparing to nitrogen and phosphorus. This is an important difference. So silicate, here I've made a statement, silicate limitation affects phytoplankton diversity but not biomass. So the point I want to make here is, when you don't have silicate, but if you have the same amount of N and P, you will still get these probably the same biomass of a phytoplankton, but the diversity changes because diatoms will be uh, restricted. Diatoms will have silicate limitation. Diatoms cannot grow. So other primary producers will dominate. Okay, so when you have silicate limitation, the phytoplankton diversity or the community profile changes, but not the biomass of the primary producers. Okay, so here we know the important differences between nitrogen, phosphorus, and silicate. So now we come to a specific problem. So normal conditions when you have your nitrogen, phosphorus, and silicate from the rivers going to the sea, you it will these will be nutrients for the phytoplankton, for the diatoms. You have your phytoplankton growth, primary production, very, very healthy environment. However, when you restrict the water flow, when you build dams, okay, either for power generation or to control floods, things change, okay? Because when you restrict water flow, the N, P, and Si that is accumulated here will be utilized by the diatoms. Okay, so you'll have phytoplankton growth here. Okay, and when you have phytoplankton growth here, although the N and P may be recycled by the bacteria, maybe degrade the phytoplankton may be degraded, and then you'll have your N and P. The silicate will sink. Okay, so silicate remains in the silica shell. So water that flows post dam, after the dam, will have the same amount, maybe the same amount of N and nitrogen and phosphorus, but it will have a reduced amount of silicate. Okay, and because of that, your the conditions at the river mouth has changed. There will be silicate limitation, and so, as I mentioned earlier, the biomass doesn't change, but the diversity, the community profile changes. You will get a different kind of predominant phytoplankton. And what we've observed nowadays is sometimes you get harmful algal blooms. You have dinoflagellates. So the population changes. You have a different kind of phytoplankton. So that is why it is important to uh, deliberate carefully before deciding to build dams, especially large dams, because large, you must remember, when you build a large dam and you impede flow, the dams will not last because siltation. A lot of the soil that is being carried in the rivers will sink and will clog up your turbines, will clog up the dam quickly. That's why most of the large dams require excavation. You have to dig up a lot of the precipitation, a lot of the sediment every 10 years. You have to dig it up. And most dams don't last beyond 20 to 30 years. Okay. The recommendation now by most marine scientists is not large dams. If you need to have dams, build small dams. Okay, because smaller dams have a smaller impact towards the environment. Okay, but think carefully first before deciding to build a dam, because the consequences are uh, long term and detrimental. Okay, for example, the Sungai Selangor Dam. when they built it, they totally didn't didn't think about the firefly community, population in downstream. So now it happens that because of the reduced flow, the fireflies are not thriving as well as before. So a lot of before building dams, you need to have really a good environmental impact assessment study that covers the whole river towards the river mouth and maybe even a few kilometers beyond to see what kind of possible effects building a dam will towards the environment. Okay, so not so straightforward.